Okay, everybody. So I'm not here today to walk you guys through the Enzyme Lab in class, but I still want you guys to be able to do the lab because I think it is a fun lab and we usually get some good data out of it. So I'm going to walk you guys through each part of the lab in this video, and then you guys will be able to complete the enzyme catalysis lab on your own without me here, hopefully with this video to guide you and with the protocol that you guys have and read and took that quiz on, right? So today for the enzyme catalysis lab, we are going to be doing 3 separate little mini experiments, although the second one is really just establishing a control for the third one. The first one is a more observational study. You guys have hopefully gotten this sheet here to start class today. This is the data sheet you guys are going to use to collect data on, so you can follow along as we go through the lab with the data sheet as well as with the protocol. So you want to have both of those out, okay? In the first, actually, before I get into the actual experiment, it is super important for this experiment, and especially because I'm not here today, that we do not have cross-contamination. So I have gone ahead and labeled everything that you're going to be using, and I want you guys to pay very, very close attention to the labels on everything, okay? You're gonna need to use catalase in pipetters and boiled catalase in a pipetter and water in a pipetter. Well, the pipetter that says C is for catalase, BC is for boiled catalase, and DW is for water. You're going to use disposable cups to run your reactions in. I've labeled all of the disposable cups. Here's experiment 1 part A, experiment 1 part B, experiment 1 part C. We've got experiment 2, which has 2 cups, and I'll talk about that in a minute. And then experiment 3, which actually has 10 cups, and I'll talk about those in a minute. But they are all labeled so that you should know exactly what goes in what cup. Additionally, you're going to use different syringes today. And again, very important, we don't cross-contaminate. We've got a T syringe that is for transfer, not titration, transfer, okay? H is for hydrogen peroxide, which is our substrate for today. Remember, our enzyme is, oh my gosh, I'm drawing a blank here. What is our enzyme? Catalase, that's right. We've got an S syringe for sulfuric acid, which is how we're gonna stop our reactions today at very specific time points. And then we've got this, it's either gonna say titration or it'll say KMNO4, which is potassium permanganate. That is for our titration, okay? Purple stuff goes in here, not in the transfer pipetter, okay? Again, I'll walk you through how we're gonna use each of those in a minute. Lastly, most of the supplies in terms of like the chemicals and things are gonna be on the sports or the table over by the sports banners. So you'll have to go over there and get most of your supplies, although you do have a couple beakers that you can fill for specific things, okay? So let's get into the experiments. Now that we know we're not gonna cross-contaminate and that everything is labeled, I'll walk you through it. In the first part of the experiment, we're really just gonna observe the enzymatic activity of catalase. And so in experiment, and you're gonna record your observations in the data sheet here in this first table. So part A, if you follow along with the protocol here, you're gonna take your hydrogen peroxide syringe in your experiment 1A cup, and all you're gonna do is put 10 milliliters of hydrogen peroxide in there. Where are you gonna get the hydrogen peroxide? Well, you have this beaker labeled H2O2, and you can fill it up with 1.5% hydrogen peroxide right here. I will have several of these over on the table. All you gotta do is fill up your beaker. You are gonna need a fair amount of hydrogen peroxide, so you might as well go ahead and fill it up all the way. I should have made plenty, so we don't have to worry about running out. Fill this up all the way, and now you've got hydrogen peroxide that you can work with, and you can fill it up later if you need more. But for part A here, you're gonna take 10 milliliters of your hydrogen peroxide. Make sure you're closely looking at that. Put it in your cup. Then you're gonna take 1 milliliter of catalase. The catalase, in the C little pipetter here, where the 1 milliliter mark is that little mark right there, right before the bubble. Catalase is going to be in a beaker. Where did I put the beaker? Let's see. Oh my gosh, can't find it. Well, the catalase is going to be in one of the beakers that you can get community supplies from. However, catalase needs to stay on ice. Okay, so what I'm going to do is I'm going to fill this thing up with ice, and then I'm going to put the catalase beaker into the cooler here. And then you guys can just come to the cooler and get 1 milliliter of catalase each time that you need it, which will be 6 times. So you'll need less than 10 milliliters, right? Just come over to the cooled catalase, cooler, get your 1 milliliter of catalase, go back, and for part A, you're going to put it in there, and then you're just going to swirl. Okay, part A, you're just going to swirl for about 30 to 60 seconds, and then you'll observe the reaction and record your observations in enzymatic activity right there. That's part A. Part B is pretty similar. It's actually almost the exact same observation, except now we're going to put our 10 milliliters of H, hydrogen peroxide, in there. Then we're going to put 1 milliliter of boiled catalase, BC, and that is not in the cooler because I have boiled it. It'll just be in a beaker sitting over there. You only need 1 milliliter of this, so it'll be a relatively small beaker where you just take 1 milliliter out. You're going to put it into our experiment 1 part B cup. Again, swirl for 30 to 60 seconds, and then write down any observations, and that will let us know what the effective temperature was on our catalase. The last one is checking for the presence of catalase in living tissue, and the living tissue that we're going to use is potatoes. Okay, so I've already started cutting these up. Essentially, you're going to get one of these little potato cubes, and you're going to macerate it. The potato cubes are actually going to be stored in this jar or in this glass dish of water so that I can kind of preserve them. I hope that doesn't influence the data. I've never done that before. So we'll see. You guys, I'm running a little experiment on you guys too. So you get your potato, you take your experiment 1 part C cup, you put it in there, okay? But we need to macerate the potato. So there'll be several of these cups over there by the supplies. You just go over and you kind of smash the potato and mash it up a little bit. Make sure you keep it in the cup, okay? So I kind of bring it up, let it drop back down, mash it up, all right? All right, there we go. Now I've mashed up my potato. That's going to give our hydrogen peroxide access to the cells on the inside, because what we're going to do next is take 10 milliliters of hydrogen peroxide and put it in here with the potato. And then we will give it a swirl again for like 30 seconds here. And then we'll see what happens. Do we see a reaction similar to up here? If so, that means catalase is probably present. If we see something like this where we might not see much of a reactivity, then that means catalase is not present. So we'll record our observations there, and we'll make some inferences and draw some conclusions about that. All right, that is part 1 of the experiments. All right, moving on to part 2 of the experiment. Like I said, part 2 of the experiment is really just setting up a control or a baseline for part 3 of the experiment, in which we are going to record the rate of the reaction at several different time periods, okay? And so for part 2 and part 3, you will be doing titrations, okay? And so you will actually need 2 different cups for each time period. So for part 2, we're just going to start with the part 2 cups here. You're going to need your catalase. You're going to need all 4 syringes, sulfuric acid, transfer, hydrogen peroxide, and potassium permanganate, okay? The way this is going to work, again, for the first part here, we're just trying to establish a baseline, and that baseline value is actually going to go in every single one of these. Okay, this essentially serves as like a control. Where are we starting at with the reaction? Okay, and so how do we calculate that baseline? Well, we're going to do a titration of hydrogen peroxide essentially without the catalase. And then later when we do it with the catalase, we'll be able to use that one without the catalase as a baseline, okay? So how do we do this in part 2? Well, you are going to take your experiment 2 cup, and that is where you're going to do your reaction. And then you'll transfer some of that to your experiment 2 transfer cup, and that is where you do your titration. And so to set this up, first thing we're gonna do is take 10 milliliters of hydrogen peroxide, okay? And we're gonna put it in our experiment 2 cup. You're also gonna need 10 milliliters of sulfuric acid, okay? And so for that, sulfuric acid is either gonna be in containers like this, or I will have some out in a beaker like this. If this gets low, you can just put more of this in there. But you'll need about 60 milliliters total in the experiment. So you'll have to go back a few times to get some, but you get 10 milliliters of sulfuric acid, get that ready to go. This is how we're actually gonna stop our reactions at very specific times. We let our reaction go for a certain amount of time, and then we squirt the sulfuric acid in there. That's going to stop the reaction because it should denature the enzyme, which prevents it from working anymore, okay? So we got 10 milliliters of hydrogen peroxide in here, Okay, we've got 10 milliliters of sulfuric acid ready to go. Next thing we're going to do for the control here is 1 milliliter of deionized water, which will also be in a beaker labeled DI water. So you get 1 milliliter, that's all you'll need for the whole experiment, and put it in there with your hydrogen peroxide and give that a swirl for about 30 seconds. Okay, once you're done, sulfuric acid goes in. We've stopped the reaction, even though there's no reaction here. Remember, this is just serving as our baseline control. And this is the important part. Now you take your transfer pipette, the one that has a T, transfer, and you take 10 milliliters of your reaction and put it into the transfer cup. This is where we're going to do our titration. And for our titration, you take your potassium permanganate, okay? And what we really want to figure out is how much potassium permanganate do we need for the titration to be complete? Because the amount of potassium permanganate needed for the titration is going to be equal to the amount of hydrogen peroxide that was consumed in the chemical reaction, okay? So again, this is dark purple material, it stains, be careful with it. You need to figure out how much potassium permanganate we use to get the titration to be complete. So you have to record an initial reading and a final reading. And when you subtract those, that'll be our baseline. That's how much potassium permanganate we needed when there was no reaction, right? And so what you need to do is fill your potassium permanganate or your titration syringe up to 10 milliliters. And then that is your initial reading, right? To do a titration, you slowly add one drop at a time while swirling the cup, okay? Until it stops being clear and turns to a light pink or a rosy gold type color, right? Make sure you're swirling it, okay? And it can't go back to being clear. Once it turns into that rosy gold color, let's say I used this much potassium permanganate, okay? That final reading for me would be at about 2.5 milliliters. Probably go to 1 decimal point here, okay? 2.5 milliliters, which means my baseline would be 7.5, okay? That's the value that's gonna go in every single one of these boxes. That's how much potassium permanganate was needed for the titration when there was no reaction, okay? So that's the end of experiment 2. We now have our baseline. For me, it was 7.5 milliliters, okay? Moving on to experiment 3. We're gonna do the same thing we just did in terms of the process. We're gonna do 5 titrations, except this time we're not using deionized water. We're actually gonna use the catalase. Why are we doing 5 different reactions? Well, because we're gonna do the reaction, we're gonna stop the reaction at 5 different times. So for experiment 3, part 1, okay, the 10-second one, you've got a 10-second cup and a 10-second transfer cup. For the 30-second, you've got a 30-second cup and a 30-second transfer cup. 60-second cup, 60-second transfer. You've got a 120 cup and a 120 transfer and a 180 cup and a 180 transfer. You do need 1 cell phone, so you can run a stopwatch here for this part of the experiment. But essentially, you're gonna do the same titration. You'll put in the 10-second cup your 10 milliliters of hydrogen peroxide. Then you will drop 1 milliliter of catalase in there, swirl for 10 seconds, and then as soon as that 10 seconds is up, 10 milliliters of sulfuric acid to stop the reaction. Transfer 10 of your reaction to your 10-second transfer cup, and now do your titration over again, where you fill it up to about 10, okay? Record an initial volume of 10, and then you do your titration in the transfer cup, okay? I'm actually gonna do it here, 1 drop at a time, all right? Then whatever your final volume is, you record here. And then to get your data, you do initial minus final. That gives you the amount of potassium permanganate used for this titration. However, we have to subtract it from the baseline, because the baseline tells us how much was needed when there was no reaction, and then we can subtract the amount used when there is a reaction, and that will actually tell us how much how much hydrogen peroxide was consumed when there was a reaction during that time period. Do the same thing with 30 seconds. So after 30 seconds, sulfuric acid goes in. After 60 seconds, sulfuric acid goes in. Every single time you transfer 10 milliliters of the whole solution to the transfer cup, and then you do your titrations. Okay. Last thing on the analysis here, you are going to graph that data. Okay. When you come back next class, you need to have your graph done, all the analysis done, so that we can discuss the data as a class. Okay. All right. That should be everything you guys need to know. I'll have everything set up and ready to go. If you get stuck at any point, this video will also be posted in Canvas in the module. And so you can revisit the video, but hopefully the protocol is pretty clear, and this video has kind of shown you how everything's gonna be done. And when I get back, we'll see what kind of data you guys have. I will be back at school tomorrow on Wednesday for the full day. I should be back on Thursday as well. My son is feeling a little bit better, but his ketones were rather high yesterday, so we're keeping him home out of an abundance of caution today, which is why I'm not here today, just like I wasn't here yesterday. But I should be back. I think he'll be fine to go to school by Thursday. So I'll be here on Wednesday, and I'll be back on Thursday, presumably, to review the data with you guys, go over our exams, and start chapter 9, right? Your homework is to finish this analysis sheet, okay? And if you have any extra time today, make sure you are reviewing chapter 8 before we move on to chapter 9, because it does get more complicated. And make sure you are working on that reassessment, which is due on the 25th. So that is only 1 week away. Okay, remember that's on College Board. So go on to College Board to our classroom and you can complete the reassessment. All right, that's it, guys. Again, sorry I can't be here today. Good luck on the lab. I'm excited to see how your data turns out.